Okay, so I want to kind of walk you through this to make sure you understand and are able to put together kind of everything we've done. So the cool thing about genetics at this point is it really starts to incorporate a whole bunch of different things we've learned. So we'll take this pea plant here, for example, and we'll say it's got, let's just say it's got a red flower, okay? Or you notice it's got green leaves, or maybe it's got a yellow seed. There can be any characteristic you want in there, but let's just stick with the red flower for a second there, or pink flower, or whatever it is. And then you have to ask yourself, okay, well, how does it know how to do that? How does it know? How does this pea plant, when it grows, how does it know how to make say a red flower or whatever? Well, anywhere in the flower, if you took anywhere in the flower and you took a sample, all living things are made out of cells. So I've sort of expanded this. This is a typical plant cell here. You can see the chloroplasts in there, which do photosynthesis. There's a mitochondria, so you should know also that plants do cellular respiration as well. So that would be found anywhere on this plant. You could take a cell. Okay, well, inside that cell is a nucleus, and inside that nucleus is the DNA. And essentially what happens is that DNA can be copied, and you can copy a piece of that DNA into mRNA. The mRNA then goes out, and then it's read by the ribosomes. And remember, the ribosome act as a functional place for the mRNA and the tRNAs to come together. That then codes for a particular protein, and then that protein then ends up forming often some kind of enzyme. And that enzyme, really, having that enzyme and that functional active site is what allows you to take certain products and either build them or break them down and so forth. So in this case here, this is actually glucose and fructose, but it doesn't really matter what this example is, but essentially the reason this flower is red or pink or this seed is yellow is there are enzymes that convert the products into yellow colors or green colors and so forth. So at the end of the day, really, what is making the pea plant become even a pea plant and one that has pink flowers, all the information, everything that this thing can develop is all coded for in DNA, which then is read by ribosomes with tRNA, and then you make these functional proteins. And so really it's the enzymes at the end of the day that are controlling all the reactions. So just to show you, Mendel found in his certain characteristics and the kind of things you got, these tall stems and dwarf stems and smooth seeds and wrinkled seeds. And what he found was typically what we call this 3-to-1 ratio. And that's how Mendel kind of figured this out. But just to show you, he didn't really get 3 and 1 every time. He would get like 787 tall plants and 277 dwarfs, or 5,474 smooth plants and 1,850 wrinkle. But what he was able to do is realize that's about a 3-to-1 ratio. This shows you some of the pea plants and how he would count those, and you could see, for example, that's a green smooth one most likely, that's a yellow smooth one, that's a green wrinkled one. Okay? Now, what happens is you have to be able to kind of figure out what's going on, and you want to be able to figure out how to build and use these things called Punnett squares. So a Punnett square basically shows you the chances of certain events happening. So what you do is, and it doesn't matter where you start or put these, but what you do is you put the different products of meiosis on one from the female. So a female gamete is basically the eggs, and the male gametes are typically like the sperm. And you put all the different chances of different things you can make on one side versus the other. And so, and again, it doesn't matter where the male or female goes, as long as you have all the options there. And then you ask yourself, okay, what if this particular pollen fertilized that egg? That's what you would get. Okay, so that's a Punnett square, and that's how you read it. So what happened in the case of Mendel was, let's say you took a plant with yellow seeds. Okay, so this is a plant with yellow seeds, but this particular one, you notice it has a big Y and a little y and a big Y and a little y. Well, that could give you then big Y, which would give you a yellow seed. Big Y, little y, which would give you yellow seed. That's another one that's the same. But notice this one here is green, because green, it turns out, it's recessive. Two little y's give you that particular one. So we're going to walk you through that here in a second. And you should realize that what happens is these are traits or sections of DNA that are found on chromosomes. So these represent chromosomes, and that yellow there is designed to represent a piece of DNA that codes for the gene that makes something yellow. So if you got this one, say, from mom, this chromosome from mom and this one from dad, and you had a copy of that and a copy of that, that's the same copy, that would give you yellow seeds. Notice over here, this one has one of each. Okay, so from your mom, which is the maternal side, you have the gene that gives you yellow, and over here you have the green, which would give you green on the paternal side. But in that case, that's what you call heterozygous, because they're different. That'll still give you yellow seeds, because remember, yellow is dominant over green. The only way you get green is when you have two green copies, one from mom, one from dad. Okay? Now, what we're going to do, we're going to take this, try to walk you through this here and show you. So let's say I have a female flower and I have a male, or I have a, because the plants actually have male and female parts on both. So I have my female here and my male over here, and I have my female being all yellow flowers, yellow seeds, and my males over here always give me green seeds. Well, when this plant, when they go to reproduce, remember that when you go through meiosis, okay, when you go through meiosis, remember, you can only give one set of chromosomes, right? So if this were a human, remember, in a diploid cell in a human, there's typically 46. Well, if you did not go through meiosis, but instead did mitosis, you would make a sperm or egg that also had 46 chromosomes in it. That would then get together with a sperm or egg that had 46, and you'd have 92. And as I mentioned before, lots of extra chromosomes, it turns out, is not good. So you have to reduce the number of chromosomes down to half. And so meiosis, that's what meiosis does. And so in meiosis, you split whatever chromosomes in half. And then what happens is half go in one and half go in the other. Okay? So you can only make a chromosome, as long as everything goes well, one set in each one. So that represents meiosis. Now, since we're only looking at yellow-seeded plants, there's not a lot of options. So since we're looking only at yellow seeds, and these are the same in the P generation in both, this is homozygous dominant and this is homozygous recessive, the options are it can make an egg, since they label this the female, we'll call that the egg, that Y or that Y. Those are your only options. Okay? So you put one Y there and one Y there. The male's the same way, but they're both lowercase because they represent the recessive allele, because this one's green. So little y, little y. And then what you do is you go, okay, what if that sperm or that pollen fertilize that egg, and you cross them? That shows you what you would get. So again, the Punnett square shows you the options of all the things that could possibly happen. So notice this is your F1. Your F1 is your filial generation, which basically means those are the offspring or the babies. The filial F1 stands for filial, and those are the offspring or the babies. So in your F1, what you get is you get all yellow, just like we mentioned with Mendel. Okay, those seeds, when you grow them, they give you all yellow seeds. Now, you can't see though that you can in this picture, but what Mendel couldn't see, because he just see the yellow seeds, he couldn't tell just by looking at them that they were different than these. They look the same because they have that one copy of that dominant gene. So what you have to do is look at what happens in the F2. So now what I'm going to do here, just to show you how that works. Okay, here's my F1 generation. Okay, so if I take that F1 generation where I had all yellow seeds, but they were big Y, little y. They were heterozygous, okay? And I grow that plant up. Here's that plant, okay? If I self-pollinate that plant, in other words, I take the sperm from the pollen, which has the sperm, from that plant and fertilize the egg from that same plant, which is another thing you can't do very well with dolphins and mountain lions, right? Is if I self-fertilize that, what I get is the F2 generation, which would look like this, where I would have this 3-to-1 ratio, where I've got basically 1, 2, 3 that are yellow, 1 that's green, and it turns out genetically two of the 4 of those you expect to be big Y, little y, and one of them turns out to be big Y, big Y. And again, all the yellow ones look the same, so you don't know, you have to grow them and see. But it turns out in your F3 generation, if you were to pick this one, it would give you all yellow seeds. If you pick either of these two, it would give you your 3-to-1 ratio again. So this F3 here, this particular one, ends up looking like the F2. So you notice those look the same. Whereas if you take this green one, you'll get all green seeds, and it'll be like the, well, I guess it's not up here, but this will be like the P generation of that particular one that we saw there. Okay, just another Punnett square to show you what you have here. Just is a little bit bigger. Here's big Y, here's big Y. That gives you big Y, homozygous dominant. Both copies are the same. Okay, now this is where it gets quite tricky, okay? So let's say now instead I cross 2 different traits at the same time. Let's say I'm looking not just at smooth seeds and wrinkled seeds, or yellow and green, but instead I go, well, what are the odds of getting smooth and yellow and wrinkled and green? Well, what you do is when you cross-pollinate those in the P generation, you get kind of what you expect in the F1. They're all 100% smooth and yellow. Okay, they're all smooth and yellow. Smooth, it turns out, is dominant over wrinkled, and yellow is dominant over green. Okay, but in the F2, when you cross or when you self-fertilize that one with itself, or if you were to cross it with something that had that same genetic combination, you would get in the F2 a 9-to-3 to 3-to-1 ratio, where 9 of them would be, and again, these are ratios, not necessarily how many you get, but you would have 9 that are smooth and yellow, 3 that are wrinkled and yellow, 3 that are smooth and green, and 1 out of 16 that are wrinkled and green. That's the famous 9-to-3 to 3-to-1 ratio of the F2 when you do what's called a dihybrid cross, and you're crossing and looking at 2 different genes at once. Okay, we're going to stop there, and I'll walk you through that on the big Punnett square on that coming up.